much for coming. Let me say that I got that, to this demonstration of something that we're calling the SPARC Dataset Viewer. This is really a tool for looking at individual data sets and tying the metadata a little bit more closely to your exploration of these different data sets. And I am giving this on the behalf of KCORE, which for those of you who are new to SPARC or may not know how it's organized, we are one of the 4 cores that make up the Data and Resource Center, and KCORE is responsible for data curation. So I know if you are a SPARC participant, you have interacted with KCORE at some point, because we are the ones who work with you to put your data into what we call the SPARC dataset structure. But I also want to introduce Jerry Skifos and Jesus Martinez of Metacell. Metacell is a software development scientific company that specializes in helping researchers develop high quality software. So they actually developed this tool in working with Tom Gillespie and others in KCORE. And so I want to acknowledge them for the work that they've done and the beautiful tool that they've produced. So just a little background, why did we go ahead and produce this tool? As you know, if you're part of SPARC, all data in SPARC is organized according to a similar structure, which we call the SPARC Dataset Structure, or SDS. And this is based on the brain imaging data structure, which has been enjoying great uptick in neuroimaging. So for those of you who might be familiar with it, but whereas BIDS is generally organized around a single modality, so MRI data sets or functional MRI data sets or any specific data type, SPARC deals with so many different multimodal data sets that we had to basically modify it so that we could handle all of the different types of data that were coming into SPARC. So there's a tremendous advantage to organizing data according to this common framework, in that if you go to a SPARC data set, everything is organized in a similar way. So that means it's easier to write tools against it and also ensures a uniform user experience. But those of you who may have worked on the SPARC portal with data sets, if you've come through the SPARC portal or when you're trying to put your data into this format, it's a little bit artificial in the way that it is organized. So basically what happens is you have different folders, and they have a lot of subfolders because we ask that you organize your data according to a folder that is named after your subject. And then we ask if that subject has multiple samples that you organize. You create a couple other folders that have the sample names. And at some point you get to the data files, and all of the metadata that describes what SAM1, SAM2, and Sub1 is are in these separate files over here. So this same name is in a spreadsheet that says this is sample 1, and sample 1 is from this particular anatomical region. So all of the information is there, but it's often very difficult if you're trying to browse or understand, either troubleshoot a data set, how exactly it's organized. So that's why I have a but, right? Understanding key details about a given subject, for example, or a sample or a file requires that you open and align multiple files. And this was the guidance that I had produced for people who were trying to work with SPARC data. So if you go to the SPARC portal, you'll see a file called subject. You should open the subject's XLS file, which is 2 directories back, and it's going to tell you what you want to know about that particular subject. And then if you open the sample, the same thing, you find a sample, and then you find files, and those files are described in manifests. So we really wanted to take advantage of the regular structure that is given to a SPARC data set to develop a tool that lets us actually browse and understand how these data sets are organized in a much more intuitive way. And as I mentioned, this was done for the curatorial team because they often have to make decisions about how data sets are organized, and they have to make sure that all the metadata is there. And this was a tool that was going to allow them to do that. But we also think that it may be useful in general if you use SPARC data, whether as an individual investigator who's preparing their data or somebody who's on the SPARC portal, to be able to understand what this data set is all about and much more easily relate metadata to, for example, individual subjects. So essentially, that is what the SPARC dataset viewer does. It's in data form. So as I mentioned, it's a curatorial tool, and I'm going to mention that several times because a lot of stuff that you will see, you'll go, what does this mean? Why are they telling me errors? Those are all things that curators need. It also, again, is not, we are planning on releasing a beta version of this onto the SPARC portal that you would be able to play with and use, but it doesn't fully work with the complete data set. So I just want to make people aware of that. But you can see here, instead of now taking a structure that is a bunch of separate folders, all organized according to these individual hierarchies, it basically presents to you the organization of a data set as a graph. So as we'll see when we actually look at this in the viewer, this basically says there's a bunch of subjects, and here are the subjects, and here are the types of data and files that are associated with each subject. You can immediately see how many subjects there are. And as I'll show you, if you click on one of these, it'll say, and this is a mouse, and it's 12 weeks old, and this is a mouse, and it is 5 weeks old, right? So it basically allows us to tie the metadata, but it provides a graphical interface that says, here's how all of the data are organized, okay? So if we look at a little bit of the interface itself, this is what the tool is going to look like. It essentially has 3 windows. The first window over here is all of those folders and files that are just now presented in a hierarchical form. But unlike the SPARC portal, where if you open the primary folder, you can't see what's in the derivative, here you can navigate and look at all of these simultaneously. So if you say, well, gee, I see that you have 5 subjects, and I would like to see if the same type of data is available for subject dorsal 1 and dorsal 4, you can just expand that out. So it makes it a lot easier to browse the hierarchical structure of the folders and the files because everything is presented to you all in one navigatable window. The second window is that graph structure that I talked about. And the nice thing about this is, again, it can give you an overall view of what this data set is about. So as soon as you load it up, and you can see that there's X number of subjects and X number of samples per subject, and here's where all the files are. But it also does something nice, in my view, that the current portal makes very difficult, and the SPARC dataset structure makes very difficult. And that is the SPARC dataset structure. Let me just go back a few slides. Basically says you can have primary data for a subject, you can have derived data for a subject, you can have raw data for a subject. And those are 3 different types. So if you go to a SPARC data set, you'll see a folder called primary, you'll see one called source, and you'll see one called derivative. If you open those up, you'll see subject 1, subject 2, subject 3, subject 4. That means that the same subject who has primary data, derivative data, and raw data, those files are in 3 different branches of the file structure. So you don't get to see what is available per subject, because sometimes a subject may have some primary data, which might be a stitched mosaic, some derivative data where it's been segmented and a beautiful 3-dimensional reconstruction has been produced, but not every subject has that, only a few of them have it. So if you look at this window here, what this one does, it says, I have a subject who's called subdorsal 1, that's up here. It has a sample, and off of that sample, there's both primary and derivative data. So it groups all of the files that are available for a particular subject or sample under one root, as opposed to the way that you browse here, which is let me open the primary folder, let me open the derivative folder, right? Let me open the source folder. So it's the same data structure, it's just visualized in a different way, okay? And I think that that's one of the most powerful things about this, is this ability to immediately look and say, as we'll see when we do it live, This one's got a bunch of files here. This one has derivative data. This one doesn't have derivative data. The other nice thing about the SPARC dataset structure tools, the visualizer, is that when you click on one of these folders, which represents a sample, or one of these folders, which represents a subject, it displays the metadata associated with that subject or sample in this window over here. And you'll notice one thing that it does. So here on this sample, it says this one is from stomach, for example. This particular mouse that it comes from is an adult male, right? And the nice thing is that it displays the metadata all the way up to the root. So instead of just looking at the subject metadata in that Excel spreadsheet or just the sample metadata in that spreadsheet, it says you have found this particular file, or you have found this particular sample, and I'm going to tell you all the way up to the top of the tree what it's associated with. And that includes if this subject is part of an experimental group, for example. It will say, well, this one is from the experimental and this one is from the control. So it gives you the metadata all the way up the tree so that you get a complete understanding of the fact that this stomach is from this particular mouse, which is from this particular experiment and this particular project, right? So you can go all the way up the tree. So I think that it is very, very powerful. Now, as a curation team, we are finding when we go through here that sometimes metadata, which we know is contained in those files, aren't there, which is why, again, it's a valuable curation tool, because it says, well, over here, it says the age of the animal and you're not showing the age of the animal, but we'll work all those kinks out, right? I think the main point about this tool is that unlike what we have now, you can go all the way up to root, and that includes file metadata, file level metadata, if that has been provided to us, right? So you can say this is particular file is this, it comes from this sample, comes from this subject, comes from this particular data set. So I hope that that is clear. Please feel free at any point to ask me any questions. The other thing that I want to say is that it is really designed right now to work with the Pensive discovery, or not the discovery platform, the Pensive platform. So if you click on any of the links up here, for example, it doesn't take you to the SPARC portal, it takes you to the Pensive version of it. But it also works on your local data set. So if you wanted to use this, for example, to download a data set from SPARC, or if you're organizing your data according to SPARC, you would be able to point this tool to your local data set, load it up and visualize it just like this, okay? So those are some of the main features. So this is sort of the demo process, the demo that I wanted to go through. So I wanted to put a slide to remind me of all the different things that I wanted to show you. But one of the things that's actually very nice about the tool is that it lets you, visualize the entire graph structure of a data set. But some of these data sets are really large. They have thousands and thousands and thousands of files. So it actually does give you an opportunity to change the layout. This is using a radial layout. And you can see, for example, that hanging off this little root here, there are thousands and thousands and thousands of files. Clearly, this is not one that you're going to want to manually browse, right? So you can use it to back out like you had in the slide that I showed you, and I'll show you in just a minute, to say, oh, this gives me the overall structure. But you can also change that layout if you get something that looks a little odd, and I will show you what that looks like in a moment. And you can say, gee, this is really too big to visualize in that hierarchical form, so I'm going to use a different layout. Okay, so let me I'll get out of my slides. There we go, and go into, hopefully, the data set structure, itself. Hey, come over here. There we go. Here's our SDS viewer. Yes, here we are. Okay, so we are now live. Scary. I see some questions in the chat. Oh, no uploading or downloading. Yes, that's correct, Dan. Right. And one thing we want from everybody is, if this is a useful thing, we would like to know how it should work. Okay. So in order to load up a data set, here's a data set that I have preloaded. So we'll go over some of the features that I just talked about. And this is the viewing mode that Anita set, right? Which is, I just want to look at what the whole data set looks like. And this tells you that here's my data set. It gives me the ID that the data set has in Pensive. If I come over here, it tells me all of the people who contributed to it. And if there's protocols and other things, those are sort of hanging off the data set level. And then it says it has a folder called subjects, okay? And that if I look at subjects, here's the name of each one. And it's got a little thing that says, well, if I want to know what hangs off of subjects, subjects have both primary and derivative data. Now, in this case, there's no data that's associated with these because they are associated at the level of the sample, but it tells you, right, that there's nothing here. You really need to go look over in the samples. And then it tells you that there is the primary data and the derivative data. And you can see that in this case, it's very well organized. Each one has these files. And if I click on one of these, right, it zooms up and that's where it tells me the metadata. So the minute I click on one of these folders, that's when it gives me the metadata. It's got this nice visualization tool. And Jerry and Jesus, if I'm missing a key feature, please jump in, but it's basically telling you the connectivity. If I click on the sample, that's when the sample data shows up, right? So here it is way down here in stomach. So as I navigate through, it gives me that additional information. Now, there actually is metadata in many cases associated with files, but a lot of times we don't get that metadata from people. And so oftentimes you look at it and it just says this is a TIFF file. But for example, now with some of the new microscopy tools, we will have things like, well, these are the proteins that have been labeled inside of a given file, for example, okay? So it makes it really easy to understand the structure of a data set overall. So how is it organized? How many subjects are there? And if I want to then browse through and say, well, here's another subject, changes it, right? Says this is male, this is adult. In this case, they're all the same, so it doesn't tell you all that much information. But if there is an experimental group, for example, it will pull that up. And again, if I go down here, it'll tell me the sample metadata. But one thing I think that is just really powerful is it taking you all the way up to the root so that you put all of this exploration in a larger context, okay?
So what if I was missing some files in some of these subfolders? How would I see that?
Well, I don't know how you'd see something that's missing. If there's something that is supposed to be there. So there are sometimes, and you will see in some data sets, question marks. And a question mark means that the tool ran into a problem and it's not able to interpret what it sees. Is that fair to say, Jesus and Jerry?
I mean,
yeah,
for files that are missing, if you don't see the icon there, that's the biggest clue.
Right, but you do get question marks, right? So the question marks means there's a problem somewhere, right?
Yeah, yes. So for sometimes like samples that are loaded but have nothing under primary or any sort of files or folders in there, like they can show up as having an issue with them. Yeah,
that's right. So
So the other thing I was just going to try to point out is that, this is a nice regular structure and there are always 5 files in the primary and 2 in the derivative, and that's a nice kind of repeating structure.
Oh, no, did I? So all you do to load it up is what we have it now. This tool is pointing to the public data. So the only data sets you're seeing are publicly available data sets. And you can see this one looks a little bit different than the other one. But again, if I can come over here and I can open up the files this way. And if I click on a subject, it'll take me to that subject. So it's simultaneous browsing. And then you see over here, it tells me that, unfortunately, this is a cat, but it's 17 months old and what its weight is. And this one in particular has more metadata that's associated with it, right? But once again, it says this one has raw data, primary data, and derivative data all associated with it. So the windows sync across them. So you can, at any given point, look at this. You can look at this and look at this. If there's a file over here, a file name is in particular, if you come across one for something, you can search for it over here and it will try to find that inside this data set for you, okay? And then if you want to reset it so that you can see the whole thing, that's when you can use this little button right here. You can pan, you can zoom, you can drag it around. And I wanted to show the radial view. That's a little button over here. So you can either use this top-down sort of hierarchical, or you can use the radial view. And if you use the radial view, it'll make a nice graph. There it is, right? And sometimes this is much more helpful. I did have a data set loaded that was extremely large, that one that I showed you, that fuzzball. Oh, actually, I can go back and get it because I put all the names. So hold on. Let me go up to the fuzzball. And this is spatially tracked. So right now it searches by ID, but I always search for it by the name of the data set because that's easier for me than those big ones. Oh, this one has a parentheses in it. So I do that, and there it is. Now, when I try to load this, it's going to take a long time because there seem to be thousands and thousands of files. But when it does load, you'll see that the main graph is really not very helpful. The main graph looks like nothing because it's trying to fit all those thousands and thousands of files there. It'll take a little time to show up because it is a very big, complex file. That's why I had it preloaded, but the best laid plans, as they say. So anyway, we'll wait for that to come up, and oh, it's not letting me go here. It's thinking too hard. So are there any questions about this while we're waiting for it to show up? Because that's basically the tool in a nutshell. Okay. Yes.
We do always have.
There it is. Can you see it?
Yay.
Yes.
So you can see that that's really not very helpful. You know, you have some navigational tools, but in this case, I find the radial is much more helpful because then it tells you why you can't see anything. And that's because it is a giant fuzzball. That will take a while. Okay, somebody had a question. Was that you, Tom?
I had a reaction, Jean.
Oh, yes.
It makes me think of it kind of makes me think about how innovation in the sort of file browsing space on our computers has sort of ground to a halt, and that we don't, because this would be incredibly useful in many contexts, not just in the scientific data context. That, you know, the Macintosh Finder or Windows Explorer should be able to do things like this by now, but no one cares about looking at files anymore, so they're probably not spending any time doing that. So I sort of think it's interesting because we used to, you know, early on in SPARC, we used to fight with people about bids, and they would say, you know, I don't want to arrange my folders like that. I want to arrange them some other way. And we would say, well, if you arrange them this way and you're consistent, then a software tool can come along and rearrange them however you want and make it look however you want. And it sort of seems like that's what this does, which is very cool.
Yeah.
So
no,
that's
exactly right.
just my reaction to it. And it's almost like if you imagine we're going to eventually get rid of folders in our file systems and everything will be tagged, then this is almost like a sort of custom way of, you know, generating a file system from a set of tags in an arbitrary scheme that, and it, you know, for maybe Marianne likes it this way and Anita likes it this other way and I like it this third way, and we can all have it look the way we want.
Yeah. And what I always like to say about visualization tools and knowledge, right? Sometimes I want to look at it this way because that's helpful to me. Sometimes I want to look at it this way, right? And at no point do I not want to know everything about this thing, right? That's ultimately what's about. This is kind of psychedelic. It's kind of cool. But, you know, and that's really the power of having this structure, is that it lets you do that, right? It lets you transform it, but you have to build the software to do that. And up until you build the software, it's a burden, right? But as you say, the payoff is that once you have something regular, then developers can develop against it. And then you end up with better tools and more flexible tools because you have this capacity here. So I think that's basically it. I mean, that's really what the tool is about. It's just much easier for you to go across and understand a data set. It was probably a mistake uploading this gigantic one because it consumes everything. Here we go. But here's the reset button, and it will take you back here. And I can just go along here and say, here's subject 1. And this is all about subject 1. And here's subject 2. And if I'm looking for something specific, right? If I know from my description that there's multiple subject groups, I can go ahead and use those. In this version, because of the way the underlying data model is done, the subject group is just sort of a field that hangs off of a subject. So if you click on a subject, it'll say this belongs to the anoxic group, for example. But in future versions of it, it will include that in the hierarchy so that you can see here are all the subjects that are gathered under this particular root. Any other things that people would like to see demoed or comments? Otherwise, what I will do is go back here and let people play with it, understanding, right, that it's beta. Okay, but what is basically next for this tool is we are just going to make it available as is in tools and resources so that people can play with it. It doesn't do things like if I click on a file, it doesn't go and retrieve it. It doesn't visualize it. Those are all things that can be done, but we'd kind of like to know whether this is going to be a tool that is more generally useful than simply for curating and maybe preparing data to put it into, or doing a quality check on what the SOTA upload tool or something like that does. So we'd really like to know what you think about the tool. There's a GitHub repository, which gives you the code and explains all about this. At some future point, it will be available through the tools and resource section. Before we go to the demo part, I think, Jill, did you put the link? Yes, there it is. Okay, there's the link. I just want to acknowledge all of the work of Tom and Jill and Kcor and Jesus and the Metacell development team. I think they did a really nice job on this tool, and I hope you enjoy playing with it.
And I'm just going to request that there's also a link to a survey form. So when you get a chance to play with the tool, if you could please give us feedback, that would be terrific.
So I think that looks like we have something that people can play with. If you have the link, you're certainly free to play. Thanks. Ask people in your group to play with it, take the surveys and get it back to us so that we can plan future development of this. And again, I want to thank the Metacell team for putting together such a nice tool from a very complex structure. It took a while to get it right. And of course, Kcor for all of the work that they do. But it's always good when one of these things comes together into something that you can actually use. So I'm very happy.
All right. Thank you, everybody.
Cool. Thanks a lot.
Thanks, guys.
Thank you.
Nice seeing you.
Yeah.
Bye.
Bye-bye.
Thank you. Bye.